Today, our lesson is on molecular basis of genetic diseases. So, what is a genetic disease? It is a disease caused by defects in an individual's genome. Do you know the most common genetic disease among Sri Lankans? It is thalassemia. What is the most common genetic disease affecting the oral cavity? It is oral cancer. So these genetic diseases can result from defects in chromosomes. These are the chromosomal disorders or defects in a single gene. These are monogenic disorders or else defects in multiple genes. These are polygenic disorders. So among these three types of genetic disorders, polygenic disorders are the most common type. All the common diseases such as diabetes mellitus and cardiovascular diseases are polygenic disorders. And these genetic diseases can be either inherited or acquired. Inherited genetic diseases are inherited from our parents. That is, the genetic defect is transmitted to the offspring via gametes. And acquired genetic diseases result from exposure to radiation or toxins after the birth of an individual. Now let's see what these chromosomal disorders are. As we have discussed earlier, DNA in our nuclei are tightly packed with the help of histone proteins and non-histone proteins to form chromosomes. If we want to observe the chromosomes in our cells, what should we do? We have to first take some nucleated cells. So we can very easily take a sample of blood and isolate the white blood cells by centrifuging the sample. Then what is the next step? We have to induce their cell division. And then we have to stop the cell division at metaphase. Why should we stop the cell division at metaphase? That is because it is the stage where the cell's DNA are most tightly condensed to form the chromosomes. Then we can stain these cells and observe under a light microscope or a fluorescent microscope. If we use a dye such as Giemsa, we can observe the cells under a light microscope. But if we use a fluorescent dye, we have to use a fluorescent microscope to observe our chromosomes. This is how we can visualize our chromosomes under a fluorescent microscope. Can you remember the specific term that is given to this kind of a picture? It is a karyotype. By looking at a karyotype, we can have an idea about the number of chromosomes, their sizes and their shapes. And also we can tell whether it's from a male or a female. Is this karyotype taken from a somatic cell or a germ cell? This is taken from a somatic cell. That is why we can see a diploid number of chromosomes. What is meant by a diploid number of chromosomes? That is, each chromosome is having two copies. One copy is from our mother and the other copy is from our father. But if we take a germ cell, they will have a haploid number of chromosomes. That is because during gametogenesis, the meiotic divisions will reduce the number of chromosomes by half. People with chromosomal disorders may have two major types of alterations or defects in their chromosomes. They are numerical changes and structural changes. Do you know what's meant by numerical changes? It means the chromosomal number in somatic cells is either increased or decreased. That is more than 46 or less than 46. If this increment or reduction occur in multiples of the haploid number of chromosomes, that is 23, the defect is called euploidy. For example, if a single ovum is fertilized with two sperms, the resulting organism will have how many chromosomes? 69 chromosomes. That is 23 from the ovum and 46 from the two sperms. So it's a case of euploidy. Since it is greater than the normal number of chromosomes in human cells, it can be more precisely called polyploidy. Similarly, if the increment or reduction in the number of chromosomes is not an exact multiple of the haploid number of chromosomes, the defect is called aneuploidy. In aneuploidy, there may be one or several additional chromosomes or one or several chromosomes may be missing. If there are additional chromosomes, the condition is called hyperploidy. And if the chromosomes are missing, the condition is called hypoploidy. For example, if we take our 21st chromosome, a normal individual will have only 2 copies of it. If somebody is having 3 copies of the 21st chromosome, it is called trisomy of the 25th chromosome. If somebody is having 4 copies, it is called tetrasomy of the 21st chromosome. And if somebody is having only a single copy, it is called monosomy of the 21st chromosome. And if the 21st chromosome is absent, it is called nullisomy of the 21st chromosome. Now let's discuss about the structural changes on chromosomes which can lead to chromosomal disorders. There are 4 major types of structural changes. There are deletions, insertions, inversions and translocations. One of the common chromosomal disorders among Sri Lankans is Down syndrome. So this is a patient with Down syndrome and this is her karyotype. What is the abnormality you can see in this karyotype? It is the trisomy of 21st chromosome. Down syndrome is important for you all because it can affect both the structure and functions of the oral cavity. So once you pass out as dental surgeons, you should be competent to rectify these problems and help these patients. Some of the structural abnormalities you can see in their oral cavities are decreased root to crown ratio of teeth, decreased tooth size, hypodontia, that is less number of teeth in the oral cavity, and delayed tooth eruption. And these patients will have problems in suckling, swallowing, mastication and speech. And there will be continuous outflow of saliva from the oral cavity, and that is called drooling. This is one of the interesting findings about Down syndrome. As you can see here, the risk of delivering a child with Down syndrome drastically increase when the mother's age is more than 40 years. That is because the rate of non-disjunction in oogenesis increase with the age of the mother. Another example for chromosomal disorders is cancer. In many cancer types, we can observe both numerical abnormalities and structural abnormalities in the chromosomes. Do you know what is shown here? This is a karyotype from a normal human mammary epithelial cell. This is a karyotype from a cell line established from a breast cancer. Can you see any difference in these two karyotypes? What are the abnormalities you can see in the cancer cell karyotype? There are numerical abnormalities in the chromosomes. Here it is tetrasomy, here it is pentasomy. And can you see any structural abnormalities? Yes. As you can see in the normal karyotype, different colors are given to the different chromosomes. So mixing up of colors in the cancer cell karyotype indicates mixing up of different parts of the chromosomes. So this is an example for the presence of both structural and numerical abnormalities in the chromosomes of cancer cells. The next type of genetic disorder we are going to discuss today is monogenic disorders. In this word, mono means single. So as the name implies, only a single gene will be affected in these disorders. In monogenic disorders, we can very frequently see single nucleotide changes. And these changes are called point mutations. These point mutations are of three types. The first type is substitutions, and the second type is insertions, and the third type is deletions. So what's meant by a substitution? It means a nucleotide in a gene is replaced with another type of nucleotide. In this case, a thymine containing nucleotide is replaced with an adenine containing nucleotide. Then what's meant by an insertion? It means a new nucleotide is added to a gene. In this case, in between the cytosine containing nucleotide and the adenine containing nucleotide, a new guanine containing nucleotide is added. And in the case of deletions, an existing nucleotide in the gene is removed. Here, the cytosine in the 6th position is removed. So, it is not found in the gene anymore. Well, now I have 2 questions. The first question is, can we detect these point mutations in a karyotype? What is your answer? Yes or no? The answer is no. Why? Because they are very tiny changes, and therefore just by looking at the chromosomes in a karyotype, we can't identify them. So please do some reference and find the molecular biology techniques which can be used to identify these point mutations. Then my next question is, how can these point mutations affect our cells? Will they always cause diseases? Okay, let's take some examples and try to find the answers to this question. Imagine that this is a sequence of bases in a gene coding for a protein, actually a very small peptide. When this gene is expressed, first it will be transcribed into a mRNA molecule, and then it will be translated into a protein. According to the genetic code, the codon AUG will be calling for the amino acid methionine, and the second codon GAG will be calling for glutamate, and the third codon UAC will be calling for the amino acid tyrosine, and the fourth codon UGA will not call for any amino acid. Instead, it will give the signal to stop the protein synthesis as it is a stop codon. So when this gene is expressed, it will produce a tripeptide containing methionine, glutamate and tyrosine. Now think of a situation where this cytosine is replaced or substituted with thymine. So will this gene mutation be reflected in its corresponding RNA molecule? Yes, the base sequence in the corresponding mRNA molecule will be definitely changed. In the normal mRNA molecule, the third base of the second codon is guanine, but now it has changed into a adenine molecule. In the normal mRNA molecule, the second codon is GAG and it has called for glutamate. It is GAG and has called for glutamate. But now it has changed into GAA. And as you can see here, GAA codon also is calling for glutamate. So in this situation, there was a mutation in the gene, that is in the DNA, and it was reflected in its corresponding mRNA molecule, but it is not reflected in the corresponding protein molecule. Why is that? That is because the mutant codon had called for the same amino acid. It is possible because of the degeneracy of the genetic code. Do you know what's meant by the degeneracy of the genetic code? It means that the same amino acid can be called by different codons. In this case, glutamate can be called by the codons GAA and GAG. Because of that, even though the codon is changed, the amino acid that is inserted has not changed. Therefore, there is no change in the protein. So this is an example for a mutation which will not give rise to any abnormality in the resulting protein. Now if we look at our second example, here thymine is replaced with adenine. So which type of a mutation it is? It's a substitution. Thymine is substituted with adenine. So what is the effect of this DNA mutation on RNA? RNA molecule also is mutated. In the normal molecule, the second codon is GAG, and in this case, the second codon is changed into GUG. Is there any effect of this RNA mutation on the protein? Yes. In the normal mRNA molecule, the second codon GAG called for glutamate. But now, in the mutated mRNA molecule, the second codon is GUG and it's calling for valine. So this will change the structure as well as the functions of the protein. Actually, this is the type of mutation present in the beta globin chains of sickle cell anemia patients. Even though it's a single amino acid change, it can alter the structure and functions of this protein. This kind of mutations are called missense mutations. That means this mutation will miss the sense of the protein. In our third example, guanine is replaced or substituted with cytosine. So what is the effect of this DNA mutation on mRNA? In the normal mRNA molecule, the third codon is UAC and it has called for tyrosine. But in the mutated version, the third codon is UAG. And as you can see here, UAG is a stop codon. So this mutant codon will not call for any amino acid. Instead of calling for amino acids, it will give the signal to stop the protein synthesis. Therefore, this type of mutations are called nonsense mutations. So, point mutations in DNA in the form of substitutions may not have any effect on the protein or synthesize a protein with structural and functional abnormalities, or else they will cause premature termination of the protein synthesis. The type of point mutation that is detected in our fourth example is an insertion. As you can see here, the normal gene contains adenine after cytosine. But in the mutant version, there is a guanine molecule inserted between cytosine and adenine. So is this mutation reflected in its corresponding mRNA molecule? Yes. There is insertion of a new base in the corresponding mRNA molecule also. This new base will alter the entire reading frame of codons during translation. So the codons before the new base are not altered and they will be calling for the same amino acids as before. Those are methionine and glutamate. But the codons after the insertion of the new base has completely changed, and now these codons are calling for a completely different set of amino acids. Even this stop codon is disrupted now, and synthesis of this protein will not be stopped at the usual place. This type of mutations are called frame shift mutations because the reading frame is altered due to insertion of new bases. And the final type of point mutations we are going to discuss today is a deletion. In the normal gene, there is a cytosine molecule before the adenine molecule. But in the mutated gene, this cytosine molecule is deleted. As in the case of insertions, this kind of mutations also will alter the reading frame of the mRNA molecule from the point of deletion. Therefore, the amino acid sequence of the protein will be altered, and sometimes the protein synthesis will continue through the stop codon. Sometimes stop codons will appear in the middle and the protein synthesis will be prematurely terminated. This kind of mutations also are called as frame shift mutations because the reading frame is altered from the point of deletion of the base. Can you guess the reason for alteration of the reading frame in the case of insertions and deletions? The reason is lack of punctuation marks in our genetic code. Now let's look at some examples for monogenic disorders. One of them is amylogenesis imperfecta. Do you know what's meant by amylogenesis? It is the formation of enamel. So in this disease condition, enamel formation will be imperfect. Then the next example is dentinogenesis imperfecta. So as the name implies, in this condition, dentine formation will be imperfect. Our next example is osteogenesis imperfecta. Do you know what's meant by osteogenesis? It is formation of bone. So osteogenesis imperfecta means imperfect formation of bones. When bone formation is imperfect, there will be malformation of our mandible, that's our lower jaw, and maxilla, that is our upper jaw. When our jaws are not properly formed, there will be abnormalities in tooth eruption and the occlusion. Then we have hypodontia and hyperdontia. Hypodontia means presence of less number of teeth and hyperdontia means presence of more teeth. Sometimes hypodontia is referred to as supernumerary teeth also. Another example for a monogenic disorder is cleft lip and cleft palate. When the left and right sides of the lip is not properly fused during embryogenesis, it will give rise to cleft lip. And when the left and right sides of the palate is not properly fused during embryogenesis, it will give rise to cleft palate. Sometimes both these conditions will appear in the same individual. I will discuss about these diseases in detail when I do the lesson on common genetic diseases in dental practice. Until then, please visit this website and find the genes which are affected in the monogenic disorders which I mentioned today. So far we have discussed about chromosomal disorders and monogenic disorders. Now let's see what these polygenic disorders are. In this term, poly means many. So in polygenic disorders, many genes should be simultaneously mutated in order to cause a disease. Some common examples for polygenic disorders are oral cancer. Of course, other cancers also are polygenic disorders, but I specifically mentioned oral cancer since it's more relevant to you all. Then cleft lip and cleft palate, diabetes mellitus and hypertension. I will discuss about oral cancer also in detail when I do the lecture on common genetic diseases in dental practice. This is the end of the first lesson on molecular basis of genetic disorders. In the second lesson, I will discuss on inheritance of monogenic disorders. Before that, please visit the website on online Mendelian inheritance of man.